Hello, everyone. First of all, I'd like to thank the organizers of this conference for inviting me to share my research. So I'm a third-year PhD student at University of Sheffield, and part of my work involves working with temperate phages and if they can be used to improve rhizobial inoculants. So the agenda for today, I will talk a bit about my context and motivation for the research, a bit about rhizobial inoculants and challenges to their use, and then if temperate phages can be used to improve rhizobial inoculants, and a bit of my research, basically two of my research questions, their results and conclusions, and a bit about my future work. So nitrogen fertilizer use is a big problem globally, especially excessive nitrogen fertilizer use, and causes a lot of negative impacts in the environment. And these impacts also include basically degrading the soil. And this map is from FAO 2021 report, which basically shows how human activities can cause strong or light land degradation, as well as how strong or light deterioration happens under low pressures or high pressure of land use. But it also shows the stable or improvement of soils under low and high pressures, which goes on to say that our soils are resilient. But of course, this resilience is variable, and that's why we see this deterioration effects. So a bit of stats, according to the Living Planet report, which came out in 2020, 52% of the agricultural land that we use worldwide is already degraded, and we are still sort of exploiting those soils. And this calls for an urgent sort of alternative switching of practices, which just don't focus on intensive agriculture practices, but also sort of focus on practices which care for the soil and the soil microbiome, not just for sustainable or food security purposes, but I think that is something which we owe to planet Earth. So one of these alternatives that I work on is rhizobia. So what are rhizobia? I know a lot of you may not be necessarily plant scientists over here, but rhizobia are basically facultative symbionts which are found in the soil. And when I say facultative symbionts, what it means is that these bacteria are usually found chilling in the soil, unless, of course, their leguminous host plant is in the vicinity, which basically means that they will form symbiotic associations with these plants in the form of these bulbous structures that you're seeing on the screen, which are known as nodules. So inside these nodules, these rhizobia fix nitrogen, specifically atmospheric nitrogen, in exchange for carbon from these plants. So since more than a century now, a lot of researchers have been working on rhizobial inoculants and potentially improving them so that they can be used as fertilizers in the real world. However, when we look at the real world, they face many challenges, and one of the challenge, or the major challenge that they face, is very variable results in terms of plant yields, and most of them, of course, tip towards lower plant yields. So what does this mean in the real environment is, and one of the major reasons for this is because when these rhizobial inoculants are introduced into the real environment, into the soils, that they have to compete with native or local rhizobial populations, which are inefficient at fixing nitrogen, but are present in the soil. And that's why these rhizobial inoculants are basically introduced at the first place, right? However, these inefficient rhizobia, although bad at fixing nitrogen, doesn't mean that they are bad at forming root nodules. And also because they are thriving and have lived since many years in the local soil conditions, they are much more adapted to the local soil conditions, and hence are much more capable of displacing rhizobial inoculant populations. Which basically translates that into, when looking at the real world, that as a farmer, if I'm a rhizobial inoculant user and I've applied rhizobial inoculants into my field, because the inefficient rhizobia have displaced these rhizobial inoculant population, I see a lower yield in my plants. So, of course, one of the Now that I've talked about the competitiveness or the lower competitiveness of rhizobial inoculants, it becomes important that we increase, or it is important to look at increasing the competitiveness of these rhizobial inoculants. And that's where my work sort of comes in. I work with temperate phages, which I'm sure all of you know what they are, but briefly sort of summarizing them is they are viruses which infect bacteria and integrate inside these bacterial populations. And whenever the bacteria divides, they divide along with it. However, whenever the bacteria is under stressful conditions, these temperate phages are known to switch into lytic mode of cycle, and they basically release into the environment. But this happens at a really low level. What it means is that out of a population of thousands of bacterial cells, only one of the individual cell will be killed to release these temperate phages into the environment, which will then infect susceptible populations which are present in the vicinity and which are susceptible to killing or lysogeny by these temperate phages. So what this means for the bacteria or for the host bacterial populations is that they are infectious at low levels to these susceptible populations, which is of course a really huge benefit for them because they provide a niche, these temperate phages provide a niche for these bacterial populations to proliferate after killing these susceptible populations. That's one of the reasons. And second reason is, of course, they also make bacterial populations resistant to infection and killing by similar phages as well as itself, giving it, again, a higher benefit of capturing the environment and sort of multiplying. So in terms of rhizobial inoculants, what this means is that if we integrate temperate phages in our rhizobial inoculants, this would mean that these temperate phages will sort of go on to infect the susceptible, inefficient populations, kill them off, and give a higher sort of benefit for the rhizobial inoculants to form nodules and fix nitrogen, which would be translated into higher yields of plants. So of course, this all looks good theoretically, but in the real world, it's really different. So that's where sort of I was interested to look at the temperate phages, whether they do increase the competitiveness in isogenic communities in lab as well as in the greenhouse. So for this, what we did is I worked with Rhizobium leguminosarum, trifoli, TRX19 strain, and I took the rhizobia which were not having the phages, mixed them with the rhizobia which were carrying the phages. So basically that makes them lysogens in a 50:50 ratio and plated them out at 4 different time points, which is 0 hours, 24 hours, 48 hours, and 72 hours. And then after these bacteria had grown, I sort of looked at their populations to look which one was doing better than the other one. So my results, so that the ratio that I, of course, talked about is basically the fitness, as you all would know. So the first column over here is my negative control, which basically means that both the rhizobial populations that were competing didn't have any phage in them, while the second column is one kind of phage that I'm working with, or I worked with, that is phage 113. And the second column basically shows the second kind of phage, which is phage 32. So these both phages are basically 45 kb big, and the 113 phage was isolated from a rhizobial strain in France, while phage 32 was isolated from Rhizobia TRX32 in New York, actually. So coming back to the graph, so anything above 1, like I described about the ratio, right? Anything above 1 was, of course, a benefit for the rhizobia to carry the phage, while anything below 1 was seen as cost for the bacteria to carry the phage. So as you can see that at 24 hours, we don't see any difference, while at 48 hours, we did see a difference, which was significantly higher in both the phages. So this was translated to 11% benefit for phage 113, while it was a 13% benefit for phage 32 carrying rhizobia. However, at 72 hours, this sort of effect ceases, and this is because because like I mentioned, I was working with same strain community. So that means most of my phage-free rhizobia were also integrated by these temperate phages at 72 hours. So after looking at this data, I found it really interesting that, and I was sort of interested in looking at what happens in the plant. So I work with clover. So instead of sort of plating these populations out at different time hours, what I did is I inoculated 5-day-old clover seeds with these treatments exactly as I described before, and waited for 4 weeks so that I could look at the populations in the rhizosphere, which is basically the root, the soil or the substrate around the root, and in the nodules. So in the rhizosphere, I saw that, again, there was a fitness benefit, but unfortunately, I couldn't see it in 113 anymore. It was just in phage 32 that this benefit was there, and it was actually seen as a 7% benefit. While in the nodules, this sort of fitness effect, which I was observing in the lab in the rhizosphere, ceased to exist. And again, part of the reason was that most of my phage-free rhizobia had also been converted into lysogens at this point. So the conclusions for these research questions, for this research question was basically that temperate phages do increase competitive fitness of rhizobia in the lab and in the plant, just in the rhizosphere though. And this sort of effect that we were seeing is more likely to be seen in the rhizosphere than in the nodules, because if you look at the soil environment, most of the competitions happens on the root surface, in this case, around the root surface as well, before the nodules are formed. So which basically means that whatever enters into the root nodules is whatever wins on the root surface. And also, second point is that the phage integrated most of the phage-free rhizobia, like I already mentioned, and hence whatever enters in at the end in the nodules is basically whatever rhizobia are carrying the phages. So the phage, again, second explanation is that the phage is infecting and integrating inside the phage-free rhizobia at a much higher rate than killing the rhizobia. We didn't have a look at it, but yeah, I have tried to make a knockout strain, which I'll talk a bit about in the future work, and hopefully we'll be able to see a sort of pure competitive advantage in that sense. So my second research question is, do temperate phages affect rhizobial and legume symbiosis? So for this, what we did is we did a biomass experiment. So we literally grew cultures and inoculated them on clover seedlings again, and waited for 4 and 8 weeks to look at what the total biomass of the plant looked like. So the treatments that we included were, again, rhizobium species. So this time we used 2 strains, that is TRX19 with and without phage, and TRX4 with and without the phage. We sort of only used TRX32 because this was the only phage that showed a competitive benefit in the rhizosphere. And we also included negative control, which basically means that no rhizobia or nitrogen was applied to the treatments, and the nitrogen control, which basically included nitrogen application. So we then measured the plant total biomass, which refers to the shoot and the root dry weight. So what we do is we dry the plant material after harvesting for 2 days at 60 degrees Celsius. And then we basically weigh the biomass of the plant. So after 4 weeks, so we didn't quite see any difference. I mean, of course, there are some differences, but then they are sort of overlapping between the treatments. So it was not a significant difference that we observed over there. However, after 8 weeks, we did see a significant difference, which was basically that the rhizobia, with or without the phages, were much more significantly higher in terms of biomass than both the controls, negative or positive. So to conclude this research question, phage presence inside rhizobia does not affect rhizobia legume symbiosis, which is a good news, because then if in the real world we we think of introducing, or even if temperate phages are present in these rhizobial inoculants naturally, that would mean that there's no negative effect to the plant yields, especially from the point of farmers. So the overall sort of conclusion from these two experiments was that even though the phage-free rhizobia were being integrated by the phages, it is not really a cost for rhizobia to carry the phages, which I think is a really important point or important observation that we made. So future work that I'm going to do is with knockout strain. So this is my supervisor, Dr. Ellie Harrison, who basically made this plasmid, which has the integration site of the phage. So because the phage that I'm working on, that is TRX32, TRX32, has the same sequence as the phage, as the bacteria tRNA leucine, and that's where the phage literally goes and integrates into. So we created a plasmid with the phage integration site so that it could be replaced inside the bacterial tRNA leucine. And we did get a knockout strain, which is susceptible to infection, which means that we do see like sort of turbid plaques when we plated these knockout strains out, but they are resistant to phage integration once they are infected with the phages. And we sort of confirmed that by PCR. So we are going to send this knockout strain for whole genome sequencing and look at whatever mutations have happened. And then we are going to basically repeat the same experiments as the competition experiment for the first research question to look at what happens when this knockout strain competes with the phage integrated or without phage rhizobia. We'll be doing it in the lab and in the greenhouse as well. So I'm quite sort of excited to see what kind of effects it has on the plants as well as on the rhizobia in the lab. So thank you for listening. And I would also like to thank my funding institutes, of course, my supervisors and my colleagues who literally helped me for harvesting the plants, which is quite a tedious job. But anyway, thank you so much for listening.
Hi, nice talk. My name is Pablo, by the way, from Singapore, National University of Singapore. Don't you think your results will be determined by the sequence of your phage, what genome, the composition of the genome of your phage? I know it's very difficult to have the core genome of a phage. We have a lot of non-annotated or unknown proteins, but in general, don't you think that will be affected by the genome of the phage?
Thank you for the question, Pablo. So my supervisor and another research technician have done some sort of work on this already. And to be honest, we have also done some RNA-seq work to see what sort of effects the phage has. But like you said, it's a bit difficult to look at the genes specifically in the phage and then sort of say what kind of effects it has. But of course, that's on the line, and hopefully the RNA-seq would give us some more information about that.
Yeah, so on the same line, you say it integrates on the same site than the other phage, but do you know if there is any trans or cis effect or regulation of other genes, for example? If you're doing RNA-seq, I think you should know the answer, right?
No, so I have just literally got back the data, so I have to sort of analyze it and look at what actually happens there. But to be honest, I don't think it does have any other effect. It just sort of integrates there and sits there, as far as I know from previous studies that we have been doing in the lab.
Thank you.
Thank you.
Take the liberty of standing here because I was wondering, sort of following on from the question that was just asked, do you ever see like nitrogen fixation genes inside these prophages?
No, we don't have them.
Okay.
Yeah, that we know for certain.
Okay,
yeah,
so there's nothing sort
yeah,
of no obvious cargo. Okay.
Yeah, exactly. So there's no regulatory protein that we have sort of seen as yet, but then again, I have to look at the RNA-seq data and look at what's present when the phage is present, and sort of this comparative analytics in terms of RNA-seq.
Great.
Thank you.
Thank you very much again, Eliza. That was a really interesting talk.